Doctora, ¿a quién le pedimos activar? Tu prendras.
¿Sí? ¿Funciona?
Aló, aló. Buenos días, amigos, colegas, autoridad sanitaria representada por el Servicio Agrícola y Ganadero, autoridades de la facultad y, por supuesto, los alumnos de nuestra Escuela de Ciencias Veterinarias. Gracias por acudir tan numerosos a esta invitación. Hoy es una mañana especial porque tenemos entre nosotros a un gran invitado. El expositor es el doctor Lucien Garigliani, profesor de Patología de la Universidad de Ciencias Veterinarias de la Universidad de Lieja, en Bélgica. El doctor Garigliani se gradúa de médico veterinario el año 2005. Apasionado por la investigación, realiza un doctorado sobre los mecanismos de resistencia frente al virus influenza y se gradúa el año 2011 también en la Universidad de Lieja. Durante su doctorado realiza diversas pasantías en prestigiosos institutos de investigación, como en la Universidad de Erasmus de Rotterdam, por ejemplo, y participa también en diversos congresos internacionales en Europa, en Estados Unidos y Asia. Desde hoy también podrá contar que ha expuesto sus trabajos en América Latina. El año 2014 realiza su postdoctorado en el Instituto de Medicina Tropical Bernhard Nocht en Hamburgo, Alemania, donde se perfecciona en análisis bioinformáticos de secuenciación, lo que le permite ser uno de los co-descubridores de un nuevo virus dentro de la familia Circoviridae. El doctor Garigliani es investigador experto en enfermedades emergentes y, como tal, jugó un rol preponderante en el estudio y en la descripción de las enfermedades de Schmallemberg y de lengua azul, luego de los episodios europeos, de los que nos hablará en instantes. Estas dos charlas serán dictadas en el idioma de la ciencia, en inglés, y les pido que no se asusten, que pongan mucha atención, iremos lento y después haremos las preguntas de rigor, así como la discusión final en español. Bueno, los dejo disfrutar de las charlas que nos trae el doctor Lucien Garigliani. Gracias.
Gracias, Tomás. So, good morning to everybody. And as Thomas explained, I'm coming from Belgium, even if my name doesn't look like to be Belgian. So I'm teaching pathology, so we are really interested in understanding the cause of the etiologies of disease. And we get a lot of cases in the necropsy room, about 2,000 cases per year. And so, of course, we have a lot of cases of emerging and new disease, and we are really interested in studying this disease. And today we'll discuss about two economically important diseases in Europe. The first one is Schmallemberg, probably not so important for you here, because it's typically a problem in Africa and Europe, but, well, let's see in the future, it probably will spread to other countries. And it's a good example of what can be done to discover and identify a new pathogen. And the second part today will be a discussion about blue tongue disease, which is well known in different countries, and to show you how the outbreak in Europe could be limited thanks to vaccination. So let's start with this Schmallenberg virus, and first a few pictures to present you the faculty in Lieja, in Belgium. So this is a typical vet faculty, with including an experimental farm to study zootechnical animals, and with several projects of expansion like this new small animal clinics like here. So what happened with Schmallenberg is that at the end of 2011, a new syndrome of milk drop and hypothermia was identified in adult cattle in Germany and the Netherlands. So it was of unknown origin, and the first hypothesis was a re-emergence of blue tongue virus serotype 8, which caused an outbreak a couple of years before in Europe. But all testings remained negative. So this is where the first cases were observed, and you can see here a small part of Belgium. So this is Germany, the Netherlands, and here is Belgium, and here France. So typically in this region. A few months later, the main German Institute for the Diagnosis of Animal Disease, which is called the FLI, used a broad-scale microarray, which is able to detect 2,000 targets, which means a lot of infectious agents, to try to identify the etiology. And it remained, again, negative. So they decided it must be something completely new and started a metagenomic analysis and discovered a new orthopinia virus, and later it was confirmed on a larger set of animals. So this is a completely new virus, at least its description is new. They published these results in Emerging Infectious Disease journal and called it Schmallemberg virus, as we will see in a few slides, because the first case was detected in the village of Schmallemberg in Germany. Quickly, the FLI transmitted to the European authorities a provisional SOP, so a standard procedure for the diagnostic of this disease, thanks to PCR. And it was extremely fast, and it was really kind of them because it allowed a quick description of the outbreak in the other countries. In December, some abortions were observed in the Netherlands in lambs with severe deformities, like this. So you can see arthrogryposis, torticollis, scoliosis, and some skull deformation like brachydactylia here. So severe deformation and muscle atrophy as well. A couple of days later, 2 of these aborted lambs were diagnosed to be Schmallenberg virus positive by PCR, and a cause-to-effect link was established between the infection by the adult animals earlier in the autumn of 2011, and then the birth of these aborted or malformed lambs. A similar syndrome was quickly identified in cattle as well. In the end of 2011, the first cases were confirmed in Belgium, very close to the German border. And the FLI announced the isolation of the virus in cell culture. Then they performed sequence analysis and found a high identity of one of the segments to a virus known as Charla, which is closely related to Acabane. So maybe you know this Acabane virus, which is well known in Asia. So this is the phylogenetic tree showing just the relationship between these two viruses among this Simbu serogroup. In January, we validated in our laboratory the same qPCR to be able to detect the disease. And just the day after, we got a case, an interesting case from the field. So a vet called us and said, okay, I got here a living calf, but with severe neurological impairment. The first hypothesis was a genetic disease, because in Belgian blue cattle we have several of these genetic diseases which can give such kind of clinical signs. But it happened that it was not a genetic origin. And this calf survived 1 week and was euthanized for human reasons. And then we performed the necropsy, and we found a severe porencephaly, which means that a large part here of the brain didn't develop, was completely atrophied, and replaced by cerebrospinal fluid. So you can see here that even with a large part of the brain missing, this calf was still able to stand and even to drink and live for 1 week. This is really impressive. Of course, we performed PCRs to check for blue-tongue virus, because we had an outbreak in 2006 and 2007 in Belgium with similar lesions, and of course for the new Schmallenberg, and it was Schmallenberg positive. So it was our first case of Schmallenberg positive calf, and this was a really interesting case because the calf was living, and well, besides this neurological impairment, it was in pretty good health. This map just shows you how quickly the outbreak spread all over Europe. So this is in January 2012, and you can see that a few months later, in April, it spread on most European countries, at least the Western Europe. So it spread out extremely fast. This is the village of Schmallemberg, which has been well known since this outbreak, and this is the first electron microscope image of the virus. So this virus is typical for the Pathogeny viridie, so really similar to Schmallemberg and Pathogeny viruses. And by performing additional genetic analysis, the people from the FLI checked the origin, the putative origin of the 3 segments, because there are 3 genomic segments, and found out that one genomic segment has a different origin, is not related to Schmallemberg. And so probably Schmallemberg is kind of reassortment between other viruses. But they also identified that the genomic sequence of this Schmallemberg virus is not new, and probably older than what we expected. And it is probably the origin of other viruses. So it might be an ancestor of other viruses like Schmallemberg. And what we do think now is that it's probably circulated for a couple of years or decades in Africa, North Africa, before coming to Europe and causing this outbreak. It was confirmed by the identification of cross-reactive antibodies in Africa in retrospective samples from a while ago. These orthopnea viruses are arthropod-borne and transmitted by culicoides species, which are small biting midges. Then they can also be transmitted vertically, so from the mother to the calf or the lamb. So this is called transplacental transmission. In spring 2012, so a couple of months after the first cases in adults, we had increasing numbers of abortions and mortalities in calves and lambs across Europe. And we had a couple of questions about the epidemiology of this virus. Which is the actual prevalence of the virus in the ruminant population? Is it a true emergence or a re-emergence? Because we have seen that it was probably an older virus than expected. So maybe in the past we did miss the diagnosis and didn't find out. So thanks to the new diagnostic method, maybe we were able this time to find it. Which is the rate of transplacental transmission? Because we just had a few cases of abortions and mortalities, and okay, are these cases the only case where the transplacental transmission occurred, or is this rate much higher with only in a few cases lesions appearing? And the main question was, is vaccine development a priority or not? So to try to answer these questions, we initiated the serum sampling in cattle in Belgium, and we obtained a lot more than 500 paired sera from cow-calf couples. So what is nice in Belgium is that in the Belgian blue breed, almost all births are due to C-section, so cesarean section. So the vet has to go to the farm, perform the C-section, and it is easy, of course, to get precholesterol blood from these calves. This precholesterol blood is really interesting because you can really see if there was transplacental infection or not. Because you know that in ruminants, the antibodies do not pass through the placental barrier. These 519 paired sera were coming from 209 farms across the south of Belgium, and we also included a couple of retrospective cases before the outbreak, so some from 2010 and from the spring of 2011, so before the first cases, to check if the virus was already circulating or not. So this is where these samples were coming from, the south of Belgium. And the result is that we had already, a few months after the beginning of the outbreak, almost 91% of prevalence in cows. Means a lot. So the spread was extremely fast in the ruminant population. And in cows, we found almost 25% of seropositive, which means that the transplacental transmission is really efficient. When we look at what is occurring during the time where the sampling was done, is that with the time you can see that the seroprevalence is increasing in cattle. But at the same time, the same seroprevalence is decreasing in the calves born from these cattle. And the reason is that probably like other viruses of the same family, like Pathogeny, we know that the transplacental transmission is impaired by the antibodies present in cattle. So in those animals that were already seropositive, the infection failed to induce a transplacental transmission. Besides, we were almost at 100%, and a few months later in 2012, sorry, we were at 100% of seroprevalence in cattle. So it is likely that in Schmallemberg as well, these maternal antibodies have a protective effect on the invasion of the fetus. We corrected the transplacental transmission by looking at what happened at the beginning, where the prevalence in cattle was still low, because after that it was artificially decreased by the existing seroprevalence. And so this transplacental transmission at the beginning of the infection was about 28%, which is similar to what was described for Pathogeny virus in Japan. Interestingly, while many viruses were exposed in utero to the virus, only a few of them developed lesions like arthrogryposis, hydrocephaly, muscle atrophy, and so on. So this is really a question. And what we think is that there is probably a very narrow window of time during development in utero of the calf where the cells, the neurons, are permissive to the infection and can result in such lesions. We had this very high seroprevalence as soon as 9 months after the beginning of the emergence. So vaccination is not helpful anymore. It's useless to develop a vaccine in this case. It was too fast, already too late. Okay, that was published as well in EID. All the sera collected before the outbreak were negative, 2010 and early 2011. Other studies which aim to search for the evidence of Schmallemberg infection in retrospective cases failed to find any positive case. So probably it's a true emergence, at least for the last 50 years, in Europe. But probably this virus was already circulating in Africa for a long time. An interesting question is, which could be the role played by wildlife in the process, especially as potential reservoir for the infection of domestic animals. And we first decided to work on red deer and roe deer because the south of Belgium contains large forest areas with large populations of these wild ruminants. And you can see here the prevalence, which is quite high in this area. About 40–50% of these deers were seropositive for Schmallemberg virus. So probably these were involved in the epidemiology and helped to understand how quickly the virus could spread all over Europe. Another example here. So this is the result of a confirmation by seroneutralization, and the more you are here on the right, the higher the titer in antibody is. And all these are positive cases, all these are negative cases. So a large part of these root here were positive already in fall 2011. So we do believe that the wild animals were exposed earlier to the virus than domestic cattle, because the seroconversion was started earlier in 2011 already. Okay, we'll also publish this. And what about wild boar? We had this question because for Akabane virus in Japan, it is well known that pig can be a reservoir and maintain the virus population and be a risk for the neighboring cattle and small ruminants. So we wondered about wild boar and did exactly the same. And we found that in fall 2010, all the wild boars were negative, and here is about the cutoff between the negative and the positive cases. And here we can see that some of them seroconverted in between. And also, so this is a large data set, you can see. Many of these wild boars were seropositive for Schmallemberg. So probably they also could play a role in the transmission in the epidemiology of the disease. And of course, these wild animals are a problem because when your aim is to try to control such kind of disease, you cannot have any effect on this wildlife. There is no control measure which can be efficient on these wild animals. What was the evolution after that? Is that the SBV seroprevalence quickly dropped in cattle population in most European countries since 2012. We had still some sporadic cases in 2012 and 13, and now the bovine population has become largely seronegative. So the herd immunity has disappeared. And we observed a re-emergence in several European countries in 2016 and 17, not so impressive as before, but still with some consequences for the breeders, like here in Ireland, for example, in 2016. And now the question is, okay, we had important economical consequence for the sector in 2011 and 12 with a lot of abortions. And now we have a vaccine. Should we vaccinate as a prevention to avoid the disease or not? And there were a couple of studies aiming at assessing the economical impact of the disease and to compare with the economic cost of the vaccination. And it appears that so far it's essentially in beef cattle and dairy sheep that it is really important to vaccinate the animals, and probably less important in dairy cattle and lamb production. So it depends on the regions in Europe, but it depends also on the willingness of the breeders to vaccinate or not, because this is not a mandatory vaccination. But it's still interesting to vaccinate the herds to avoid the economical losses. What about the pathology now? So I already showed you this picture: impressive muscular atrophy, arthrogryposis, torticollis scoliosis, and sometimes deformation of the skull as well. So this is an extreme case, of course, and have plenty of cases in between. Sometimes only some scoliosis, but the rest, you can see here, the muscular development is normal. Here, severe torticollis. So you can see there is a continuum and a large set of clinical presentations. Couple of neurological lesions here, porencephaly. Sometimes it can be as severe as this, which is called hydranencephaly. So the brain has been completely replaced by cerebrospinal fluid. Okay, so deformation of the jaw, scoliosis, deformation of the spinal cord here, and often you have this atrophy of muscles and replacement by adipose tissue. Again, severe lesions and development defects of the central nervous system. A typical feature of Schmallemberg positive cases was the atrophy of the spinal cord. And you can see here the difference between the control, a normal calf, and here, so it's at the level of the occipital foramen, the difference here with the complete atrophy of the spinal cord. And we tried to understand how this major deformation occurred and what was the link between these neurological lesions and the apparition of this severe deformation. Here again some pictures with cerebellar atrophy, some inflammation. Most cases fail to show any microscopic lesions. But in LAMS, we have the chance sometimes to get still some inflammation, satellitosis, chromatolysis of neurons, necrotic neurons. Here, atrophy of the ventral horns containing the motor neurons in the spinal cord. Lesions in muscles, so with atrophy, replacement by fibrous tissue and adipose tissue, some mild inflammation as well. This is really the most striking lesions. This is a normal case. You can see here the spinal cord with the horns containing in the gray matter the neurons, and here you can see the atrophy of the spinal cord. And this is really typical. And we had the clinical scoring related to the deformation, the level of deformation and malformation of the animal. Again, a negative control showing normal neurons here, and here in the same area, disappearance of the neurons. And we could establish a link, a direct link, between the disappearance of spinal neurons, so we perform counting, and you can see here in normal neurons the count, and here in severely affected calves, the disappearance of these neurons in the same area. And again, if you look at the muscular lesion, we also had a scoring, and you can see that this scoring is proportional to the loss of the spinal neurons. So there is really a cause to effect link between the disappearance of these neurons and the appearance of these muscular lesions. This is the same picture, but in the publication. We also measured and estimated the ratio between the spinal cord and the occipital foramen, and you can see that also this ratio is directly correlated to the grading of the clinical lesion observed. Regarding the viral distribution in the cases, in the aborted or the young calves and lambs with mortinatalities, the virus was mostly present in the central nervous system, especially the spinal cord, which was really the place where the highest titers were observed. So, after all these experiments, we have a pathogenesis in 3 steps. During the infection in utero, the virus is able to invade the central nervous system and to destroy the developing neurons. This destruction of neurons, especially motor neurons, induces a denervation atrophy of the skeletal muscles, which impairs the movement of the limbs and induce secondary bone deformities. So this is really the pathogenesis of this virus. And this has been demonstrated later in mouse models of the disease. Okay, so we'll also publish this. So in conclusion about Schmallemberg, it has recently emerged in Europe, but it's not a new virus, probably much older than expected. It is responsible for febrile milk drop syndrome in adults, but its economical impact is mostly related to the abortion and mortinatalities. The pathogenesis and lesions are very similar to those of other autopenia viruses, like Akabane. And we know that for Akabane virus there is an endemic situation in Japan, so we expect such an endemic situation to occur in Europe as well. the vaccination is efficient to prevent the transplacental transmission, but during this first outbreak, the spread of the virus was so fast and so quick that we had no time to prepare a vaccine. But if such an outbreak was to appear again, we will be ready to vaccinate quickly to avoid this quick spread. And definitely there is a role of wildlife as a reservoir of the disease, which complicates the control of Schmallemberg infection. Thank you for your attention. I will be happy to answer your questions.
Las preguntas las pueden hacer en español, lento, de forma bastante técnica, él lo puede comprender, y si no, aquí haremos el esfuerzo por traducir todas sus dudas.
You can also ask it in English.
También pueden ser, por supuesto, en inglés o incluso en francés. ¿Por qué no?
O en francés. Yeah.
Okay, I have a question about the seasonality of the outbreaks. Are you relating the outbreaks with the rise on the culicoides species? Are you doing surveillance on the culicoides populations?
Yeah. Very good question. So typically this is a vector-borne disease, so it's highly dependent on the presence of competent vectors. And we will have this discussion about Bluetongue because the situation is the same and the vectors are the same. It was really interesting, this Schmallemberg outbreak, because these culicoides, as we will discuss in the second presentation, are mostly spread thanks to the wind. And it was so fast, the spread in 2011 and 12, because of the wind condition, with a lot of violent winds, which pushed the culicoides over large distances. And I could see in some farms that half of the animals of the farms showed plenty of abortion and multinatalities, and the other half not, just because the places where these animals were grazing in the summer were exposed or not to the winds. So definitely the presence and the distribution of the vectors are extremely important. And the weather conditions play an important role, as we will discuss in the second presentation. So yeah, there is no continuous monitoring of the culicoides species regarding the transmission of the disease because it's too complicated. But definitely the emergence of such an arthropod-borne virus is directly correlated to the level of infection and presence of these vectors, definitely.
Have you tried like traps for the farms to diminish the amount of culicoides in the area to try to?
Yeah, again, we will have this discussion in the second presentation. These techniques have been proposed and works in laboratory conditions, but on the field it's extremely complicated. Yeah, so it's like in horses which are allergic to these culicoides. It's extremely hard to get rid of them. There are always some of them, and well, just one bite is enough to transmit the virus. So it's not easy. And well, the main problem with Schmallemberg is that it was so fast. We had no time to do something. As you have seen, in a couple of weeks after the first cases, we already had the diagnosis. So I think it was really efficient. It was a quick answer. But yeah, we were just spectators and couldn't do anything. And after a few months, all animals had seroconverted. At the end of 2012, the vaccine was available, but 100% of the ruminants were already seropositive. So it was useless. Yeah.
I have a question about the current distribution of the disease. You told that it was mostly in Western Europe. And what about Eastern Europe? Have you seen anything about that?
Yeah, so I don't know exactly the precise distribution of the virus, but it has spread also to the Eastern Europe, several countries, but there are still many countries which never had the infection. Well, the main problem is that most of the ruminant population now has become seronegative, and we do not know why. We have just some sporadic cases appearing including in Belgium recently, and not a new outbreak. But we are expecting this to happen. But we are ready, so the vaccine is ready, and in case it starts again, we will vaccinate largely. Yeah.
In Spanish.
Por favor.
La importancia de la enfermedad ahora es baja, por lo que estoy viendo, ¿no es cierto? Hubieron muchos casos al inicio, algunas pérdidas económicas de alguna importancia, pero con la vacunación ya se logró controlar y ahora la enfermedad está como localizada a unos puntos, algunos casos, ¿no? Es más o menos eso lo que ocurre, y la importancia de la enfermedad no sería tan importante ahora.
I think I asked for a translation.
Alors, tout au début, la maladie a fait des très problèmes économiques.
Oui.
Mais maintenant, il veut savoir quelle est l'importance et comment ça a descendu, et c'est en relation à quoi qu'il y a les plus grandes pertes économiques.
Toujours par rapport à Schmallemberg?
Oui, par rapport au Schmallemberg, oui.
So, yeah, indeed, at the beginning it was mostly related to this abortion and mortalities. We had important losses, especially in small ruminants. So in lambs, been in some farm, it was completely crazy. Half the animals showed dystocia and difficulties for lambing, and they have several losses of ears as well. So not only the mortalities, but still also the adults, because you had this dystocia, and sometimes it was not possible to save these animals. So there are really important losses for some exploitations. Now the economical impact of Schmallemberg is almost nil, because we have just sporadic cases. There is still an impact because of the surveillance program, because we still continue to survey, and of course it means money. And these sporadic cases are still a problem, so we do not know. What we fear is that in the future we will have, such as Akaben in Japan, an endemic situation with always new cases, sporadic cases appearing, and so it's an additional burden for the breeders. You know, there are already, I think, enough problems and difficulties for the sector in Europe, and this is something more, which means several losses and yeah, some efforts to try to control disease. So the current situation is that no, we have almost no cases, just few sporadic cases, so the economical loss currently is quite low. But as we will discuss the second presentation, the overall impact is much less than blue tongue, definitely.
Bueno, elementos importantes que él acaba de mencionar tienen que ver con, bueno, principalmente no hubo vacunación porque el virus se transmitió demasiado rápido por el viento, y que es cierto que transportó los culicoides en algo entre 6 a 9 meses. Toda Europa occidental estuvo afectada por la enfermedad. Eso, y que los casos ahora son esporádicos y que principalmente los gastos están asociados a la vigilancia que hace su servicio agrícola ganadero.
¿Y la pauta de vacunación que aplican?
La vaccination, est-ce que vous faites, est-ce qu'il y a des protocoles?
Si. So the vaccination has been shown to be efficient. So as soon as you have neutralizing antibodies in the mothers, there is no transplacental transmission. So this is really efficient. So you need a protocol of 2 injections, and it's really efficient and relatively long-lasting. So it's really a good protocol. The vaccine is not expensive. It's a classical inactivated vaccine, but still it means some cost if you have to vaccinate all animals. So currently there is no vaccination because there is no need for it. But at least we are ready, and if required, we can vaccinate the animals.
¿Sí? Bueno, yo creo que se entiende bastante. De todas formas, insistir que él comenta que la vacuna sí funciona, que es altamente eficiente principalmente inhibiendo la transmisión, ¿cierto?, vertical. El protocolo son 2 inyecciones, que la vacuna no es muy cara, es una vacuna inactivada que funciona bastante bien. Por el momento no lo están vacunando porque no ha sido necesario, ya que estos anticuerpos neutralizan, tienen un efecto bastante importante. ¿Sí?
Una consulta sobre la presencia de la familia de culicoides. ¿Estos estaban presentes en esa área anteriormente? ¿Lo habían identificado? ¿O estos se han venido adaptando por el tema de cambio climático, por ejemplo?
Si, qu'est-ce que vous connaissiez avant pour la présence ou pas de ces culicoïdes, ces vecteurs? Est-ce qu'ils sont apparus aussi comme le virus émergent ou ils étaient déjà là? Qu'est-ce que vous connaissiez?
For Schmallemberg, these vectors were already present. Yeah. So we did not expect these vectors to be competent for such a virus because they are not for Pathogeny, for instance, but they appear to be completely competent and able to transmit the disease.
Los culicoides ya estaban ahí, eran competentes para poder alojar este virus y transmitirlo, pero no estaba presente la patología todavía.
Sí.
I have a question. Maybe thinking about the possibility of the disease happening here in South America. What factors do you think could influence? Maybe migratory birds? Have you any data?
I would say.
Excuse me. Esa pregunta está súper interesante. ¿La podría hacer en español también? Por favor.
Ya. Claro, pensando en quizás qué factores podrían influir en la posibilidad de que esta enfermedad ocurra acá en Sudamérica. No sé si tienen alguna información respecto a las aves migratorias, por ejemplo.
So, as far as I know, there's no migratory route pathway between Europe and South America for the birds. Definitely for several diseases it has been shown that these birds played an important role, like influenza, for instance. We have new flavivirus outbreaks in Europe because of these birds coming from Africa, along with viruses and transmitting them into Europe. But for this specific virus, I think that for Schmallemberg, and it could be the same for bluetongue, the main threat is through the airways and through, I would say, the ships. So these ships with large containers can contain cavities, or when they transport the tires, you know, for recycling, you can have some small quantities of water in these tires, and it has been shown for culicoides and mosquitoes that can allow the transmission all over the world. So I think the main threat is there. As we will discuss in the second presentation, we do not know why the virus emerged there in the middle of nowhere in Germany, close to the German-Belgian border, because it's, you know, it's not in the south of Belgium, it's not close to Africa, it's just, it appeared there, we do not know why. And as we will discuss, it was the same for bluetongue serotype 8 in 2006. It appeared exactly at the same place. We had some emergence of other viruses, human viruses, in large European harbor, like Rotterdam or Hamburg, for instance, because of this tire transport and recycling in Europe. Yeah. So this is definitely a problem. Yeah.
I have a question. Do you think in other wild animals like your reservoir, like como reservorio, otros animales?
Like which one? Quels pourraient être les réservoirs in wildlife?
Les autres que.
Ou le répéter aussi.
So yeah, we just looked for wild ruminants, the most important one, which are red and roe deer in Belgium, and wild suids, so wild boar. We do not know for the other animals. It is, I would say that many animals are exposed to the bites of these culicoides midges, and probably many more species did seroconvert during the outbreak, but we do not know currently if they are sensitive, susceptible to the infection or not, or if they can play a role as a reservoir. What we know is that mice are not susceptible. So we had mouse models, but we had to use, I would say, interferon negative mice, which are highly susceptible to plenty of infectious disease, but the wild mice were resistant to the infection. So probably the rodents are not so susceptible. But it would be interesting to screen for this to be sure that other animals could not play a role. I think there is some knowledge about it for Pathogeny, but the main reservoir for some strains, at least of birds, are suids, so the pigs. And We also showed, I didn't show this here, but we had the same kind of result for domestic pigs as well.
For bats?
I don't know. Actually, I don't know. It's an interesting question. So the bats can be, of course, kind of a prey for these biting culicoides. So maybe they could be exposed to the disease. They can also be exposed by eating these culicoides because they are insectivorous. Actually, we don't know. I don't think it has been checked so far. I told you that we had an outbreak of a new flavivirus now in Europe, which is called Uzutu. It is really closely related to West Nile. This virus is transmitted by mosquitoes, and there it has been clearly shown that bats are susceptible. And we could show that bats eating infected mosquitoes developed encephalitis and die from the infection. So they're really susceptible to the infection. But it has not been shown for these viruses, either Schmallemberg or Bluetooth, to my knowledge. Okay. ¿Hay otras preguntas?
Bueno, si no hay más preguntas, sí, por favor.
¿Se ha descrito algún caso en humanos?
No. So far there were no cases reported in humans and no seroconversion. We had, at first we thought that it was the case also in dogs, because we had a large study in Belgium looking, because we have plenty of samples of dogs, and they were all seronegative. But after that, I don't remember in which country, but they found seropositive dogs, but no clinical signs. So just an exposition to the virus, but no disease. And so in humans it's clear, there's completely no risk. Yeah?
¿Los pulícidos solo actúan como vector mecánico o también hay una...?
No, they are amplifying vectors. So not only mechanical vectors, but they are really amplifying the virus. Yeah. So they are true vectors. And for some of the viruses of this family, they are the only way the virus can be transmitted. And it is required, if you just take the virus from cell culture and inject it in the animal, it's not enough. The passage through the vector is mandatory. This is not completely true for Schmallemberg, but for other viruses of the same family, this is the case. You really need to go through the vector and get amplified in the vector before the vector bites and transmits the disease.
¿Más preguntas? Bueno, si no hay más preguntas, le damos unos 5 minutos de descanso al doctor para que pueda tomar agua, descansar un poco. Y después volvemos con la charla ahí sobre lengua azul. Así que démosle de todas formas un...